Hi, welcome to my poster presentation. Me and Christopher England did this together. Sorry if my voice kind of phases out sometimes because I'm a little sick right now, so I'm sorry in advance. But we did it on water and bacterial findings in the environments throughout the semester that we found and the introduction. So throughout the semester we have studied many viruses and bacteria, and OMZ is an important part in the carbon and nitrogen cycle. Tropical North Pacific was the main OMZ that we studied. Over the years, new technology has been invented to help better study and understand the viruses. But an example of a new machine would be the large microscope that we used at LSU about the third week, that we used to find the number of bacteria and viruses in a single cell. And we are still scratching the surface of the potential information we could gather about bacteria and viruses. So the main question that we were trying to figure out was, what are some of the different viruses and bacteria that are found in oxygen minimum zones, aka OMZs? And the results we gathered were the enumeration data showed the abundance of viruses and bacteria in the LSU lakes and ETNP. The ETNP data was provided by ETNP 6 and 2 and showed a larger abundance of both bacteria and viruses due to the greater abundance of marine life. The LSU lake showed a larger abundance of viruses, as would be expected, as viruses are much smaller and typically more abundant in marine habitats. The morphology data collected from offshore and nearshore locations show the breakdown of the structural makeup of the viral population in those areas. These areas from the ETNP data are taken from oxygen minimum zones, which are areas of the ocean with the lowest oxygen saturation. Morphology data showed a statistically insignificant difference in viral structure at depths higher than 130 meters. Lower than the viral breakup becomes less various and starts to become dominant by non-tailed morphotypes. The morphotypes were assigned by observing a microscopic photo of each virus, which could lead to human error affecting the results. The AMG data also came from offshore and nearshore samples, and the total AMG was calculated at 5 separate depths. The AMG data showed the nearshore was much higher than offshore counts, and each peaked at about 100 to 300 meters. One of the greater limitations of this project overall is the data self-collected in lab, as the level of human error is much higher than in a professional lab. The potential for incorrect calculations or misleading of equipment that could lead to false data is too common to ignore. The project's results show how the population of viruses and bacteria can add to the oxygen saturation in their local ecosystem. This data comes mostly from the ETNP and locally collected water samples. The abundance of viruses and bacteria and their components can lead to differences into the oxygen and saturation of the OMZs and potentially other marine ecosystems. And the methods we use is all on this graph right here, which I'm sure you will see whenever you look at the poster yourself. And we also use this in the final draft of our paper. And our conclusions was that the AMD data showed offshore held the more abundant in the mixed layers, and we concluded that bacteria was not as abundant as viruses in single cells, and more non-tail viruses were present than any others. DNA allowed us to see if the viruses were capable, but we were not 100% sure about it. This is helping us move forward because we are getting an abundance of collected data by all of our students that participate in the labs. And this translates to real-world events because it gives us insights on how much OMZs are different and what others are like. The significance of this project was to learn more about oxygen minimum zones and the bacteria located in it. Thank you.